Dear colleagues, I will talk about microorganisms of deep subsurface, experimental and bioinformatic tools which we used to study them. At first, a brief introduction about these environments. Deep terrestrial and marine subsurface is the largest ecosystem in total volume. According to different estimates, the deep subsurface contains between 10 and 20% of the whole microbial biomass. Subsurface biosphere goes deep up to several kilometers and is limited mostly by the increase of the temperature with depth. It's an extreme ecosystem due to high temperature and pressure and also due to low availability of nutrients, since it's thought to be isolated from the surface. It is supposed, but it's not proven, that the rates of microbial metabolism are very slow and the generation times could be up to hundreds and thousands of years. It is also supposed that some, but not all, deep subsurface environments could be isolated from the surface for dozens of millions of years. The energy and nutrient sources could be different and depends on the origin of the rocks. In the magmatic rocks, there is low organic and all microbial life is driven by hemolytic autotrophic processes. On the contrary, the sedimentary rocks contain buoyant organics, matter which could provide substrates for heterotrophs. The best known examples of such ecosystems are oil reservoirs. Here, microbial life could be much more abundant and diverse. Okay, now I will try to explain why we need molecular genetic methods to study microbial communities of the deep subsurface. Classical microbiology, like botanical zoology, deal with organisms grown in laboratory, so-called cultivated species. However, already first studies of natural environments aimed to amplify 16S RNA gene sequences directly from total isolated DNA revealed that usually less than 1% of microorganisms detected by 16S RNA sequences represented cultured species. Moreover, about half of bacterial phyla even now lacked cultivated representatives. Phylum is the highest taxon, like mammals, birds, or fishes among eukaryotes, for example. In botanics and zoology, any new species is a great achievement, but in microbiology, you don't know 99% of them. This cultivation-independent molecular studies of microbial diversity started in early 90s, caused a really revolution in microbiology. Here you can see results of just one study of microbial diversity in a hot spring in Yellowstone Park. All phyla labeled in blue were already known, and all new phyla, which were detected in just one study, are labeled in red. Most of them are uncultured until now. We only know that they exist. The question is how to study them. The only approach is genome-based analysis. And of course, most of cultivation efforts were focused on few environments and groups of microorganisms. Primarily, it's medically important human pathogens, soil microbes, and biotechnology-relevant microorganisms. In case of deep subsurface, there is a minimal number of cultivated species, and there is no real alternative to genomic approaches. Okay. There are two main approaches for genome analysis of uncultured microbes, metagenomics and single-cell genomics. In case of metagenomics, you just sequence total DNA isolated from environmental sample, assemble contigs, like in case of usual genome sequencing, and then, using bioinformatic tools, distribute these contigs between bins representing different microbial species. The result is so-called metagenome assembled genome, representing a consensus sequence of closely related natural, I would say, strains. In the single-cell approach, you first isolate individual cells using cell sorter. Then you need to isolate DNA from a single cell and to amplify it to make enough amount for sequencing. Then this DNA is sequenced like usual microbial genome. Both approaches have their advantages and disadvantages. In case of metagenomics, the main advantage is scalability. You can sequence more and you will get more and more genomes. And you will be able to get even minor community members. The problem is in contic binning, I mean misbinning. It's problematic if you have related but different species. Their genome features will be very similar and bioinformatics algorithms will not be able to distinguish them. The obtained genome could be chimeric. And it should be noted that these genomes represent consensus sequences. I mean that if you have, let's say, 10 strains, you will get something like average sequence, and you will not see variations between these strains. In case of single-cell genomics, you will get a genome at a strain level with no problems of contamination with sequences of our microbes. Of course, if you perform separation of cells rather well. But the problem is in incomplete amplification of a genome. If some part of DNA was damaged during isolation, it will be lost irreversibly. You can sequence as many as you like, but you will not get it. Usually single-cell genomes are incomplete. Only rarely you can get more than 70% completeness. Usually such genomes are 10 to 50% complete, rarely more. But you can sequence many single cells to compensate these problems. Okay, so here you can see a history and statistics of metagenomics and single-cell genomic studies. Metagenomics was the first. The studies started about 20 years ago, but for many years they were rather rare due to high sequencing cost. Single-cell genomic studies started about 10 years ago and quickly outcompeted metagenomics because they need only standard equipment, I mean cell sorter, and not too much sequencing. But recently the trend switched again in favor of metagenomics because sequencing became cheaper and cheaper, and especially due to the appearance of third-generation technique. I mean Pacific Bioscience and Oxford Nanopore. They enable generation of long reads that simplify assembly of long contigs and consequently their binning into metagenome assembled genome. Now metagenomics is clearly a prevalent approach and single-cell sequencing is used only in very specific cases, I would say. Okay, so here you can see some scientific results of application of metagenomics and single-cell genomics in environmental microbiology. The first is so-called microbial dark matter project. Here, about 200 single-cell genomes from 29 uncultured prokaryotic lineages were sequenced, and the work is published in Nature. You can see its title is Insights into Phylogeny of Microbial Dark Matter. Microbial dark matter, it's this uncultured prokaryotic lineages. There are also large-scale studies in metagenomics. For example, in this work, 8,000 of genomes were assembled from different metagenomes. First known genomes were obtained for 17 uncultured bacteria and 3 archaeophyla. The most recent international metagenomic project was published just a few months ago. 52,000 genomes of microorganisms were obtained from metagenomes. They represented about 12,000 new candidate species, and this set of genomes expanded the known diversity of bacteria and archaea about 50%. As you can see on the map of the samples, Russia, like in many other studies, is almost a white area. But still there are a few spots that my team contributed to this project. Okay, now, after this introduction and explanation of methodological details, I would like to come back to our studies of a deep subsurface. The object of studies of my team are the deep subsurface aquifers in Western Siberia. This region is known for its huge underground oil and gas reserves, but besides oil, there are many subsurface water reservoirs located at depths of several kilometers. If there is a hydrocarbon reservoir located below a sea rock layer, then the gas will be on the top, followed by oil and water in the deeper horizon. The oil was formed from marine sediments of Jurassic period, and the aquifers probably originated from the same time. The oil production in this region began about 60 years ago, and many oil exploration boreholes were drilled at that time and later. Some of them reached not all, but water reservoirs. But river water flows out under natural pressure, delivering underground microorganisms to the surface. We studied several such artesian boreholes. This oil exploration borehole was drilled 50 years ago from sedimentary rocks until Paleozoic basement. I will not go deep into technical details of drilling and testing of a borehole, but the summary is that the water originates from about 2 kilometers deep aquifer located in Cretaceous sediments. Water flows out already 50 years. It's anoxic with a temperature of about 45 degrees that will match the depth of 2 kilometers. Its total mineralization is about 2 grams per liter, and the salts are of relict ocean origin, mostly sodium chloride. This conclusion is based on chloride to bromide ratio and overall salt composition. However, most of water is derived from meteoric recharge of the aquifer, since only 5% of original marine salinity was obtained. So it was not fully conserved for 100 million years. Notably, the concentration of sulfate and nitrate are low. The main dissolved gas contained methane, and according to isotopic data, it is mostly of biogenic origin. The water also contains dissolved hydrogen sulfide, and this is an indication of a microbial sulfate reduction in the reservoir. Okay, we analyzed the composition of microbial community of this water by sequencing of 16 sRNA genes during 2 years. The community was stable and contained bacteria with a minor fraction of metagenomic archaea. The most abundant microbial groups were firmicutes, it's about 1/4 of the community, and delta proteobacteria about 20%. Both groups were mostly represented by non-sulfate-reducing species. It's the Sulforudis, the Sulfatamacrum, and the Sulfobacca among delta proteobacteria. Most of our bacteria are phylogenetically distant from known species and could be classified only at the phylum or order level. Only 30% of 16 sRNA sequences could be assigned to cultivated genera. Among abundant groups, we found bacteria of phyla Gneiobacteria, Chloroflexi, and Aminicinatus. We sequenced a metagenome of this community. Here I would like to go a bit into technical details and talk about bioinformatics. For metagenome sequencing, we usually use a combination of Illumina and nanopore techniques. Illumina generates many high-quality but short reads, and it's relatively cheap. Nanopore reads are much longer, up to dozens of thousands nucleotides, but the quality of sequences is very poor, and it's more expensive. Most importantly, in our case, you need much more DNA for nanopore sequencing, and it's consumed in one run. Concentration of microorganisms in the subsurface water is usually low, and we need to filter, I would say, 50 up to 100 liters to isolate just 1 microgram of DNA for nanopore sequencing. So it was not always possible. Anyway, if you manage to isolate enough DNA, we used both techniques. If we have only Illumina reads, then quantic assembly is done using megaheat. If we have both Illumina and nanopore, we used hybrid assembler Metaspades, and alternatively, in case of high coverage by nanopore reads, we'll use Fly to assemble only nanopore reads and then correct sequencing errors by mapping Illumina reads to nanopore contigs using Pyro. This is the best but really affordable variant. The next step is binning of contigs into metagenome assembled genomes. It is based on different frequencies of tetranucleotides in contigs of different microorganisms and coverage and some other features. Binning works well with long contigs, let's say longer than 5 kilobases. That's why nanopore is important. We are using 2 programs, Concoct and MetaBat. I can say that one is better. The outcome depends on particular metagenome, so you need to try both. Okay, the goal is always to assemble high-quality genomes. Ideally, each has a complete circular chromosome. But this is a rare case. Although bioinformatics tools became better and better from year to year, still there are some problems. Maybe some of you will solve them, at least some of them. In my studies, the metagenomic DNA is usually a limiting factor, and the most frequent case is a high coverage by Illumina rates, more than 100, but low coverage by nanopore rates, usually less than 10. So we need a program that can join Illumina contigs and fill in the gap using a low number of nanopore rates. This program must build consensus sequence of a gap using all available nanopore rates and subsequently correct the sequence using Illumina rates. This is not a trivial task, and bioinformatician who will make it will be acknowledged by many experimental researchers in the field of metagenomics. Okay, so let's go back to our results. We obtained 25 genomes with more than 90% completeness. Altogether, we accumulated 90% of a whole metagenome, so we have all information for understanding of microbial life in the subsurface. Quantitative estimation of the completeness and contamination of genomes is based on the presence or absence of a set of about 120 conservative single copy marker genes. If a sample genome contains all of them, then it's likely complete. If, for example, 90% of markers are present in 2 or more copies, then 10% of genome is represented by contigs from other organisms. Usually only metagenome assembled genomes with more than 90% completeness and 10% and less than 10% of contamination are considered as high quality, appropriate for analysis. It's important to note that there is no rule that genomes of abundant lineages are well assembled, while genomes of minor members couldn't be obtained. And if you have at least 20 times average sequence coverage, you can get good assembly for everything. The main reason of poor assembly is the presence of closely related but not genetically identical species in the community. In this case, both contig assembly and binning became problematic. This is an illustration. The most abundant genome, the sulfobacter, you can see it here, number 47. It was assembled into 32 contigs, while complete circular genome was assembled for bacteria which share in the community was 40 times less. This is number 40. Okay. This is one of the most important results of the study. We were lucky and obtained complete circular genome of a member of a candidate phylum BRC1, although it accumulated less than 1% of microbial community. Phylum BRC1 was identified more than 10 years ago by 16 sRNA sequences. They were detected in various aerobic and anaerobic ecosystems, but initially it was found in rice soil, and thus it was called bacterial rice cluster, which means BRC. It's still uncultured, and only few incomplete genomes are known from metagenomes and single cells. We performed phylogenetic analysis of our genome using concatenated conservative marker genes and found that it's clustered with 4 partial BRC1 genomes known from single cells, and all these groups form a phylum level branch in the tree of life. Analysis of a complete genome of this bacterium allowed to predict its main metabolic pathways and lifestyle. At first, we found a set of genes of hydrolytic enzymes that could degrade polysaccharides. For example, complete ketene degradation pathway is encoded, which is shown here. Sugars may be fermented in Embden-Meyerhof pathway to make acetate, and several cytoplasmic hydrogenases can make hydrogen. In addition, we found complete tricarboxylic acid cycle and all components of aerobic respiratory pathway. NADH dehydrogenase, succinate dehydrogenase, alternative complex 3, and cytochrome C oxidases. This bacterium could also perform anaerobic respiration since it has nitrate reductase and molybdentarine reductase that could reduce sulfur compounds. Overall, BRC1 bacterium could be considered as metabolically versatile organotroph. As I told, there are several other draft genomes of BRC1, but only one is near complete. It was also recovered from groundwater, and we found similar metabolic characteristics such as aerobic and anaerobic respiratory chains. Particularly, it has the same hitinolytic pathway. The presence of chitinase was rather unexpected for deep subsurface organisms because there is no obvious source of chitin. In nature, it's a mainly component of shells of shrimps and insects. We made synthetic genome with chitinase, expressed recombinant enzyme, and analyzed its activity and found that it is indeed a chitinase. It was most active at 50 degrees. That is exactly the temperature in the subsurface. So it's, let's say, highly likely that BRC1 degrades chitin in the aquifer. Later, I will talk about the origin of chitin. Another interesting organisms belong to the candidate phylum Riflebacteria. It's a genetically isolated division, first described in 2016 in the aquifer in Colorado, in the United States. Only a few draft genomes are known, all obtained from surface sites, and none was analyzed. Here we can see the reconstruction of metabolic pathways of this bacterium. Like in BRC1, we found genes encoding carbohydrate active enzymes, but not so many. It seems that the new bacterium can use only a limited range of polysaccharides, such as starch and chitin oligomers. Contrary to BRC1, this bacterium lacked pathways for aerobic respiration. Pathways for dissimilatory reduction of sulfide, nitrite, nitrite, arsenate, and sulfur compounds also were not found. But the genome included several multi-heme C-type cytochromes linked to a membrane. This is typical for dissimilatory iron reductases. And our conclusion is that Riflebacteria are specialized organotrophs capable to utilize some carbohydrates performing fermentation or dissimilatory iron reduction. The third rather abundant and interesting uncultured lineage found in the subsurface water was the candidate phylum Aminosinantes. It was discovered 30 years ago in the thermal spring in Yellowstone Park in USA among 12 first described candidate phylum. Later, this lineage was detected in various terrestrial and marine ecosystems. Until now, there is no cultured isolates, but several metagenome assembled draft genomes and single-cell genomes were obtained. According to genome analysis, Aminosinantes is a metabolically diverse group. Some members can use proteins and amino acids as a substrate. This is the reason for the name Aminosinantes. We ferment them under anaerobic conditions. In our genomes, the presence of aerobic respiratory chain or Wood-Lundgren pathway of carbon fixation was reported. Okay, so we assembled a near complete genome of Aminosinantes bacterium and reconstructed its metabolic pathways. Contrary to previously described proteolytic Aminosinantes, this bacterium encodes enzymes for utilization of various carbohydrates: chitin, starch, cellulose, mannose, galactose, and different simple sugars. Fermentation pathways were found, but no aerobic respiratory chain and Wood-Lundgren pathway. The only terminal reductase for anaerobic respiration was nitrite reductase. Most likely, this bacterium is specialized on destruction of complex polysaccharides and fermentation of sugars under anaerobic conditions. So this way, we analyzed the genomes of all dominant community members. And this enabled to obtain the main results of the first part of this work, an ecological model of a subsurface aquifer. It's quite different from deep oligotrophic subsurface environments that harbored only hemolytic autotrophs. There are several observations which need to be explained in the model. The first is high abundance of sulfate reducers at low concentration of sulfate. The second is wide range of enzymes involved in degradation of plant polysaccharides. And the third is the presence of aerobes in the community. This is now a model. The key issue is that the subsurface aquifer is not like an underground lake or water bubble, I would say. But it's a complex system where different parts, different chemistry and microbiome composition. Only at the borehole outflow, when we collected water for our analysis, we got all microbes and chemicals mixed together. The local concentration of sulfate can be much more higher in the pore water of rock matrix due to oxidation of sulfur-containing minerals of the solution of sulfate-containing minerals such as gypsum. The second source of sulfate is the connate water of relict ocean origin. So this is the place for sulfate reducers. The second point is finding of enzymes involved in degradation of plant polysaccharides and chitin. The explanation is that buried organics originated from marine sediments of Mesozoic era, mostly formed by algae. But shells of shrimps were also buried and now serves as a source of chitin. An intriguing issue is the presence of oxygen in gas samples and bacteria with capacities for aerobic respiration. It's unlikely that oxygen is a result of contamination of our gas samples because it was found in gas collected at our boreholes in Western Siberia during several years. So this is a general phenomenon. Most likely oxygen was delivered to subsurface aquifer with meteoric recharged water. And this is consistent with the data that contribution of relict marine water is only 5%. So 95% was entered into the aquifer during 100 million years. So microorganisms are probably also not randomly distributed in the aquifer. Oxygenated part, which is shown here, could harbor aerobic heterotrophs such as chloroflexi, bacteria, BRC1. Fermentative bacteria degrade organics and produce substrates for sulfate reducers, which inhabits sulfate-rich zones. We studied several such deep subsurface aquifers and always found rather complex microbial community. In another borehole, in addition to sulfate reducers, fermentative bacteria and aerobic heterotrophs, we also found many methanogenic archaea. Probably in this case, an aerobic zone of the aquifer consists of two parts. The first is sulfate-rich zone, which is shown here in yellow, and also a large part of sulfate-poor zone. It's located somewhere here with methanogens. Okay, so I will finish with ecology, and now I'm going to the second part of my talk. It will be about molecular biology and microbial evolution. One of the interesting findings in our deep subsurface aquifer was detection of a rare and unusual bacterium called Desulforudis aldaxviata. Initially, it was found more than 10 years ago in fractured water collected at 3 kilometer depth in South African gold mine. In this environment, only this bacterium was detected, nothing else. So it was a single species ecosystem. Phylogenetically, Desulforudis belongs to Firmicutes, the order Clostridiales. The complete genome was assembled from environmental DNA sample. In the South African gold mine, this bacterium has everything required for growth. According to genome analysis, Desulforudis is an anaerobic hemolytic autotroph capable of hydrogen oxidation coupled to sulfate reduction. And also it can perform carbon fixation via the Wood-Lundberg pathway. Hydrogen probably originates from radiolysis of water and sulfate from oxidation of pyrite. So it's a self-sufficient ecosystem and nothing from the surface is required. It will live in the deep subsurface for unlimited time, even after the end of all life on the surface of the Earth. So the paper about this interesting bacterium was published in Science in 2008. Besides South African mines, 16 SRNA sequences of Desulforudis have been detected by molecular profiling in several places around the world, but all of them were related to deep subsurface. It's a deep groundwater in Portugal, the geothermal water in California, and 2 industrial places in Europe where water also originates from deep subsurface. Therefore, it was very interesting to find Desulforudis in Siberian aquifer. We assembled complete genome of Siberian strain of Desulforudis from Metagenomic and found that it was very similar to South African genome. Structural differences were minimal, deletion of one prophage, translocation of mobile elements, but no acquisition or loss of metabolically important genes. When we compared nucleotide sequences of genomes of Siberian and South African strains, we obtained even more striking results. The genomes are 2.3 megabases in size, but differ only in 753 single nucleotide polymorphisms. Of them, 700 are clustered in 6 genome regions, and 31 was randomly distributed across the genome. Only these 6 regions were shown here in black. They resulted from horizontal gene transfer from related species, and 31 true SNP reflected mutations accumulated in course of vertical evolution after separation of South African and Siberian populations. It was also interesting to compare CRISP loci of South African and Siberian strains. This is one of the most quickly evolving regions of bacterial genome, and acquisition of new spacers in CRISP arrays reflect the history of interaction of bacterium with phages. And surprisingly, one CRISP array was completely identical in 2 genomes, and only one specific spacer was found in the second corpus. Such similarity indicates identical history of interaction with phages of both South African and Siberian strains. But phages are always supposed to be specific to a particular ecosystem. That means that all this history occurred before separation of South African and Siberian desulfurosis. Okay, now we wanted to estimate the divergence time between South African and Siberian strains. For such calculation, you need to know the rate of accumulation of mutations per year. This is very difficult to measure for microbes. But there are some estimates. In the paper published in PNAS, people compared genomes of different strains of thermophilic microorganisms, Sulforubus islandicus, isolated from geographically isolated geothermal volcanic sites. Since the times of formation of these sites is more or less known, it's possible to calculate the average rate of nucleotide substitutions per year. It appeared to be about 4.6 10 in minus 9 degree, something around the universal rates estimated from comparisons of other species and predicted from laboratory determinations of mutations rate in sulforubus. If we will apply this value to 31 true SNP in the sulforubus genome, we will get the divergence time of only 3,000 years. This is very striking. If we will assume that South African and Siberian populations remain isolated since their formation, this is really nothing, because the continents were split more than 100 million years ago. However, there's one questionable point in these speculations. We detected 31 SNP in Siberian genome, but this is a consensus sequence obtained from Metagenomic. So there are 2 variants. The first is that Siberian population is genetically near identical. In this case, in each cell, there are indeed 31 SNP relative to South African genome. The second variant is that Siberian population is genetically heterogeneous, with 31 SNP in most genomes, plus many SNP in individual genomes that were collapsed to consensus sequence in course of contiguous sampling. I mean, for example, 10,000 SNPs in each Siberian cell, but in different positions. To answer this question, you need, and of course, in this case, you have absolutely no estimations of divergence time. To answer this question, you need to sequence cultivated isolate or single cells from Siberian strain. We did both. Our finding promoted a large international project on sequencing of 120 single-cell genomes of the sulforudis from South Africa, North America, and Siberia. The results were recently published in ISME journal. You can see the paper, which is called Evolutionary Status of Deep Subsurface Microbial Lineage. Okay. All three sites were deep subsurface water, but they were characterized by different physicochemical conditions, for example, temperature and water chemistry. Also, they harbored different microbial communities. So, the expected hypothesis was that evolution of isolated populations of the Sulfurudis involved genetic diversification and adaptation to local environments. It is exactly what Darwin showed for birds on Galapagos Islands. But this was not observed. The average nucleotide identities of all pairwise genome comparisons exceeded 99.5%, and it didn't correlate with geographic distances. The difference between genomes from two South African sites were higher than, for example, between North American and Siberian sites. Of course, we checked that SNPs are not sequencing errors, but true polymorphisms. As a control, several single cells of bacterial cultures were sequenced, and they appeared to be much more identical. We also found no evidence for unusually strong purifying selection. DNDS values for Sulfurudis were higher than in most other microbial populations under strong purifying selection, such as, for example, marine cyanobacteria. Another confirmation of genetic similarity of Siberian and South African isolates was obtained upon cultivation of the Sulfurudis from Siberian site and sequencing its genome. Our collaborator from Tomsk State University, Dr. Olga Karnachuk, obtained pure isolate. This is certainly not a trivial task, since nobody during the last 10 years after discovery of the Sulfurudis managed to cultivate it. The genome of isolate appeared to be almost identical to genome assembled from metagenome. So the population is genetically near identical. All conclusions about genome similarities of Siberian and South African Sulfurudis were confirmed. Interestingly, Siberian strain can grow with a doubling time of 28 hours. Certainly, it's not thousands of years, as it was suggested for deep subsurface microbes. Okay, so the extremely high genetic similarity of the Sulfurudis from deep subsurface site at 3 continents is an established fact. What could be the reason? There are 2 possible explanations. The first is a fast global dispersion between subsurface sites via aerial transport. And the second is very slow involution in the deep subsurface biosphere. Let's start from the first one. There are several arguments in favor of this explanation. The first is ability of the Sulfurudis to form spore. It's a firmicute, like bacillus. Such spores could be distributed for long distances. There are examples of finding spores of thermophilic firmicutes from geothermal sites in cold marine sediments. Also, the deep subsurface sites are not fully isolated from the surface, really. There is an inflow of meteoric water, at least on Siberian side. The outflow also exists, for example, via artesian borehole in Siberia. Perhaps there is something similar in South African sites. But there are also opposing observations. The first is the absence of long-term tolerance to oxygen, both of vegetative cells of the Sulfurudis and spores. When all 16 sRNA reads of the Sulfurudis in all databases originates from deep subsurface sites. In spite of huge amount of data from marine environments and soils, no the Sulfurudis 16 sRNA were found in such environments. And the last is the data that groundwater age in some of South African sites is up to 20 million years, which is much more than what spores can survive. The second hypothesis is very long evolution in deep subsurface biosphere due to extreme genome stability. There are different mechanisms that could contribute to such stability. The first is long-term dormancy in the form of spores. But this was not supported by several observations. Active transcription of the Sulfurudis genes in South African site was shown, and in situ turnover time of biomass estimated from aspartic acid racemization was less than 1 year. And the Siberian population contained actively replicating cells of the Sulfurudis. The second is strong purifying selection. But this is not the case because the NDS values were higher than in most other microbial populations under strong purifying selection. And the third option is inefficient DNA repair mechanisms and polymerase fidelity. However, there is nothing specific in DNA repair genes. An intriguing hypothesis was that the sulfurudis has extremely precise mechanisms of DNA replication. Our American collaborators obtained recombinant DNA polymerase and found that its fidelity is not very high. It's a bit more precise than Tucker BST polymerase, but less precise than PFU, for instance. Maybe there are some unknown mechanisms contributing to precise replications, especially in vivo, but we don't know at present. But anyway, the sulfurudis presents a stark contrast to the current model organisms in microbial evolutionary studies, which often develop adaptive traits over very short periods of time. And something like a living microbial fossil that underwent only minimal evolution since the breakup of Pangaea more than 50 million years ago. Okay, now I'm going to conclusions. The first is that microbial communities of Western Siberian deep subsurface aquifers comprises methanogenic archaea, sulfate reducers, anaerobic fermentative organisms, aerobic heterotrophs, and anaerobic respirers in different combinations. Probably the availability of buried organics, spatial heterogeneity of Western Siberian subsurface aquifer, and the long-term inflow of meteoric water enabled coexistence of these different metabolic groups of microorganisms. So the deep subsurface ecosystems harbored novel uncultured prokaryotic lineages, expanding our knowledge of microbial diversity. So the hunters for new microbes will have a lot of gain. And the sulfurudis autoxylata populations from 3 continents are very close genetically, indicating very slow microbial evolution in deep subsurface or existence of mechanisms of fast global distribution of subsurface microbes. So, and the last slide will be about my team. It's the Department of Molecular Biology of Microorganisms at Research Center of Biotechnology of Academy of Science. It's Vitaly Kadnikov, Andrei Mardanov, Alexey Beletsky, and me. And of course, I wish to thank my collaborators, Dr. Olga Karnachuk from Tomsk University, Eric Beckraft and Ramonas Stepanauskas from Bigelow Laboratory for Oceanic Science, and all team working on single-cell analysis of the sulfurudis. Thank you for attention.